Hello everyone, my name is Giulia Debiasi, and in this video I'm going to briefly present you our recent publication in the Journal of Magnetic Resonance Imaging entitled Mapping Intracellular Volume Fraction with Susceptibility Source Decomposition as a Marker for Tissue Cellularity. For further details, please refer to the full article. Pathological changes in the brain often affect tissue cell composition and density. This can happen, for example, for Alzheimer's disease, Parkinson's disease, multiple sclerosis, amyotrophic lateral sclerosis, and brain tumors. In each of these examples, proxies of tissue cellularity might be quantified and monitored to inform clinical decision-making. This can be achieved non-invasively with MRI, and specifically diffusion techniques such as DTI and NODI have been employed. In particular, the neurodensity index derived from the NODI model has been applied to assess microstructure and cellularity in the brain. The recently developed Decomposition algorithm for separating subvoxel magnetic susceptibility sources provides a new tool to study tissue microstructure. Decomposition models the signal originating from three magnetic susceptibility compartments and estimates their volume fraction, namely C positive, C negative, and C0. The aims of the present study are then first to propose a susceptibility-derived cellularity index called hDCI as a marker of tissue cellularity based on the parameters estimated from Decomposition, and second, to evaluate the proposed hDCI by performing a comparison with the neurodensity index derived from the NODI model. For this work, two population cohorts were enrolled, including healthy participants and patients with glioblastoma. In addition to conventional MRI sequences acquired in the clinical setting, a standard multi-echo gradient recalled echo sequence was acquired to quantify magnetic susceptibility and apply the Decomposition model. NDI was instead estimated on images collected with a standard sequence for DTI. Anatomical images were bias field corrected, the brain was extracted, and registration to the T1 weighted image was performed. FreeSurfer was used to obtain region of interest for the analysis, and an automatic software was used to segment the tumor tissues. Diffusion volumes were instead processed with MR-Tricks, including denoising and correction for Bézierian homogeneities, eddy currents, and motion. Then the NODI model was fitted and NDI was estimated. As for magnetic susceptibility quantification, state-of-the-art processing steps for quantitative susceptibility mapping were executed. First, a brain mask was created. Then, Laplacian phase unwrapping was performed, V-sharp was used for background field removal, and ILSEQ algorithm was chosen for dipole inversion. Finally, Decomposition was run to obtain model parameters, among which the volume fractions of the three compartments that the model describes, which are constrained by the equation showed here in the slide. Given that C0 represents the extracellular fluid, by computing 1 minus C0, we obtain a measure of the intracellular volume fraction of each voxel that we call hDCI. Several brain regions and subregions of the tumor were considered for the statistical analysis, which included correlation analysis between RI means of hDCI and NDI for each cohort separately, and Bland-Altman analysis to assess the reproducibility of the measurements. Let's take a look at what hDCI looks like. Higher hDCI values were observed in the cortical gray matter compared to white matter, with elevated values also evident in the deep gray matter structures. The cerebrospinal fluid appears dark with values close to 0. Moving to C negative, for example, the maps show high myelinated structures such as optic radiation and in the internal and external capsule, while the C-positive map highlights iron-rich structures such as caudate putamen pallidum, red nuclei, and the substantia nigra. A first visual qualitative comparison between hDCI and NDI in the healthy participants shows that NDI highlights white matter and more closely resembles the C-negative map, while hDCI shows a different contrast between gray and white matter. Moreover, boundaries of the deep gray nuclei are more defined in hDCI, and the common NDI artifact related to the CSF is not present in the hDCI map. Moving to the patient cohort, it can be observed that χDCI shows a complex tissue heterogeneity within edema, that is the hyperintensity region delineated on FLAIR, while all other images appear generally homogeneous, including NDI. Significant correlations were found for white matter and putamen in both cohorts, while for the patient cohort, additional significant associations were found for pallidum, thalamus, and edema. Blood album analysis suggested that χDCI is less dependent on the acquisition protocol and the MRI scanner, resulting in high correlation and negligible bias for χDCI. Therefore, χDCI is proposed as a potential marker for tissue cellularity, and the analysis that was carried out confirmed that it represents the intracellular volume fraction of a voxel. Significant and non-significant correlations were found between χDCI and NDI across brain regions, suggesting in fact that the two metrics could provide complementary information. Furthermore, the NODI model was specifically introduced for NDI to map neurites, while χDCI might be more suitable for mapping tissue cellularity throughout the whole brain. To conclude, potential advantages of χDCI include a faster acquisition sequence and higher spatial resolution. Additionally, χDCI might be more suitable to map cellularity in neurological conditions, such as in brain tumor, suggesting tumor infiltration where other MRI images appear generally homogeneous. Finally, in combination with other metrics computed with decomposed QSM, χDCI could aid the characterization of neurodegenerative diseases, in which magnetic susceptibility and the tissue cellularity are affected by pathological changes. Thank you for watching this video, and if you have any questions, please feel free to contact me or Professor Giulia.